So this is the liver. It's almost the holy organ for biochemists. Everything happens there biochemically. And the functions of the liver include making things like clotting factors and proteins, excreting stuff from the body like bile, and also metabolism. So a lot of fat metabolism, protein metabolism, and carbohydrate metabolism occur in the liver. Now, when doctors order liver function tests, we're not usually measuring any of those functions, and what we're measuring is damage to the liver. Now, the liver cell consists of a nucleus in the centre, which mission control for the cell, and a whole lot of organelles which keep the machinery for the cell, like the mitochondria and smooth endoplasmic reticulum, and the edges of the cell, which help to export and import stuff into the cell. Now, inside that cell are enzymes. Enzymes like, which we're talking about, gamma-GT or GGT, which is on the surface of the cell and actually helps stuff get into bile. ALT is inside the cytoplasm, the main compartment of the cell. And AST, on the other hand, which is another thing that we can measure in the laboratory, is inside the cytoplasm as well as inside the mitochondria. There's sort of an extra amount of AST. These are the things that we can measure in the blood. And why do they appear in blood? Because whenever the cells are stressed, they blister or bleb and release their contents into the body's fluids. And so the level of the liver contents that appear in the bloodstream reflect a damage or turnover of the cell. And the level in the blood depends not only on how quickly it's coming in, but how quickly we're removing those things from the cell, from the blood. Now, with AST and ALT, those enzymes which are in the cytoplasm and mitochondria, when you first damage a cell, there's more AST in the cell than ALT, and so the AST levels are higher. As the damage eases off, the AST disappears faster than the ALT, and it may look like the ALT is higher, but it's actually because it persists longer. And then as things improve and the AST is removed very quickly, the ALT might look like it's the only thing that's elevated. So in the laboratory and in general practice, we should be looking at this relative ratio of the AST and ALT. If the ALT is higher, or the de Ritis ratio is low, it shows that it's a chronic or resolving condition. Whereas if the AST is higher, the ratio is high, then it shows that it's an active condition or something that's likely to lead to permanent damage. And so in the UK, they recommend that laboratories should always report those two enzyme levels. That ratio is very important in telling you whether something's wrong and whether it's going to be getting worse.